Welcome to this workshop to present the topic of translational endpoint in autism as part of this IMI third call for proposal. And I think that personally I'm very pleased that IMI is now embarking in having a topic on this subject, because there is a very big unmet need out there in Europe and out of Europe, and a lot of patients that are just now starting to be recognized that they are patients actually. Until recently, autism and autism spectrum disorder was just considered a handicap. That was, you were born with that and that's it. But now things are changing, progress in academia is occurring, and we are learning more about it, and the pharmaceutical company also are becoming aware of this important unmet need and starting to work on that. So I think that this is a very perfect opportunity for IMI to do something hopefully important and innovative with an impact for the industry and for the patient in Europe. And just the usual introductory things, just to remind everybody that this section is video recorded, so if you don't want your faces, please cover yourself now. But instead, if you want everybody the people that also can't have the opportunity to come here to take advantage of your question and the answer that your question will receive, please try to speak clearly in the microphone so that both your question and the answer will be able to be reproduced in a good way. And also I would like to stress that this section is aimed to be a very section to answer scientific question, really specific on the topic, and not then on question more broad on application procedure of IMI, on rules. We have also an information booth downstairs, and you will find a lot of information also on our website, or you can also, there will be some contact information, you can send us a mail or phone us. So the topic will be presented by Will Spooren from Hoffmann La Roche. And just to give you a brief introduction on Will. Will is studied biology and graduate in the field of neurobiology at the University of Nijmegen in Holland, and received his PhD at the Department of Neuropharmacology and make his postdoc training in the same university, and then at the University of Rochester in US. And then he joined industry, Novartis, where he lead a behavioral pharmacology laboratory. And there his main assignment was elucidation of pharmacology of metabotropic glutamate receptor with emphasis on group 1 and 2 receptors. And his publication in this area indeed have become seminal papers. In 2001, he joined CNS research at Roche, where he continued his effort in drug discovery, particularly neurokinin and glutamate receptors. And in 2008, he joined the CNS Translational Medicine Group as R&D team leader, and there is responsible for project in early clinical development, which is up to phase 3, and is author of more than 60 peer-reviewed papers and patents, and is an elected member of the board of the European Behavioral Pharmacology Society, where he holds the position of General Secretary. In 2008, together with Roche colleague, he received a research prize from the German Fragile X Society for the discovery of phenoblam, a selective mGlu5 receptor antagonist. And in the same year, he also was winner of the Roche CEO Award for Innovation and Excellence. In 2010, he received the Pioneer Award from the Fraxer Research Foundation for his pioneering work on mGlu5 receptor antagonist. So I think he's an expert on this field, and I think it should be a pleasure to work with him. And the presentation of Will be again repeated kindly by Will as a webinar on the 19th of November. And there will be full information on how to access this webinar on the IMI website. And there will be the presentation again during the webinar, and there will be also the possibility to make questions using a chat function. Now some just very nitty-gritty practical information that are aimed to try to help you to make a good expression of interest. First of all, really in general, you will see, and this information is provided in the guide for applicant, that there is a template for preparing your expression of interest, and this template is divided in different sections, and it's very important that you follow this template and you try to fulfill and address all of this section, simply because for AOEs to be eligible to be sent to evaluation is a criterion that all these sections are filled. So please be careful about that. This is a very technicality, but it's very important. And also what is important is that you will do your submission using an online tool. And this tool will allow you to save your submission, but then at the end will also ask you to finalize this submission. And it's very important that you do this last step, because if you don't do this last step, the submission will not get to IMI. So it's not only important to save, but also to finalize. So now, after this technical sort of information, from the more scientific point of view, and also to prepare for the presentation of Will, I would again recommend that you read very carefully the topic text, which has been really, let's say every word has been really, is important. And you will see that there are a series of scientific objectives that are addressed in the topic. It is important that you consider all of these objectives in your expression of interest. And you will see also that there is a suggestion for how to organize the work in order to deliver this objective research as work packages. And this is a suggestion of the topic writers. But if you have some good idea and some innovative project design proposal that you think are even better to deliver this objective, you are very welcome to propose this design of the project. But what is very important is, in any case, it's that you keep in mind that you as applicant are expected to make key contribution on the defined variable and all of them, and that this contribution will have to be combined with the work of the EFPIA consortium to really create a collaborative research effort. And you will see that there is also some information on which we'll elaborate more on what is EFPIA going to contribute for this joint adventure, and that this contribution is mainly in-kind contribution. And, but on the other hand, this is not something that is really fixed, because this will be refined and discussed more once the winner consortium will be selected and will meet with the EFPIA consortium, and they will have to discuss and work together in order to create a full project proposal. And then finally, just to say, if you need more information, or you have a question, or you are in doubt of something, you are very welcome to contact me. This is my email, or send a mail to Infodesk, or also, of course, search on the web of IMI. But please do not contact the EFPIA coordinator, because in this period from now until the end of the submission, they are not allowed to talk with the applicant for obvious reason. And then for what concerns looking for partners, we have a partner search tool. Here you see the URL, how to get to it. And there is also one of our colleagues downstairs that is there really to explain how to use this tool. And now I would leave the word to Will.
Thank you very much, Elisabetta, for this nice introduction. So I will be giving you a flavor of what this call topic is about, how we define it from the EFPIA side. I'm the project coordinator of this project, and I will speak on behalf of EFPIA, but I didn't come alone, so we have Claudia Pop here, who is the project manager. Can you stick up your hand? We have Franco Borsini from Sigma Tau, and we have Rob Ring from Pfizer here. Can you? Hey, good. So in total we are 6 companies, and we have very strong EFPIA consortium. I think you will see that later. So we'll talk about a little bit about autism, and you probably know that a lot more about me, because although I may know something about autism, if anything, I'm an M2-5 receptor antagonist specialist, so I have a lot to learn as well. But autism is really an area that is defined by 3 core symptoms: impaired social interaction, repetitive behavior, and impaired communication. Now, there's also a substantial set of comorbid phenotypes that include, among others, depression and anxiety, irritability, cognitive impairment, and OCD. What is important, however, for the field is that we realize now that about 1% of all children are being diagnosed with autism. The size of that population, that incidence, is really the same as it is for schizophrenia. Now, Big Pharma has obviously been traditionally very active in these areas, but has now realized that this is an area of considerable concern to the population, of a high medical need, but above all, and that what we'll see later, we now understand much more of the underlying biology, which will allow then drug discovery. So the prevalence rate is also very increasing. It is about 10 to 70% annually, and there's no obvious explanation for that, but probably that has to do with the awareness of the disease and better understanding. Treatment is relatively erratic in the sense that there's no drug developed for autism specifically, but there are various drugs that are being used in this population. Not so much on evidence-based treatment, but more on trial and error. No medication to date is available for the core symptoms, and this is obviously a very high medical need. And as I said, for a drug company, in order to develop drugs that are effective, it's very important that we have evidence-based therapy, and therefore we need to develop a context which is currently absent in Europe. So there are 2 key developments in the field, which I will briefly highlight, that has developed over the course of the past 10 years, I would say, but were really accelerated in the last 2 to 3 years, really. So that's how new this is. And one of this is the fact that we have identified, and various genetic consortium have done a brilliant job here, identifying multiple genes that somehow provide a susceptibility factor for developing autism. Now, this is really a massive improvement, and this is a totally different situation what we have with, for example, anxiety, depression, or schizophrenia. Now, identifying that genes is one level that can provide us some insight. What is the underlying biology of autism? But it's really how these genes come together. And that's what happened within the field. And what we have seen is that several forms of monogenetic, as well as some other forms of autism that have been linked to certain genes, come together in the synapse. And the synapse seems to be a key structure that provides somehow a susceptibility for developing disorder. What is important as well is that mouse models have been generated, and they seem to confirm what we're seeing in various cellular assays, and that the behavior and synaptic physiology underline the importance of these proteins. Furthermore, there are common biological pathways for brain development and plasticity now identified, and I think we will see more of that in the near future. So we look on the graph on the right. They're basically, we had a telephone conference last Tuesday with the EFPIA consortium, and we were saying there's basically 2 different pathways, maybe 3. That's the P10 mTOR pathway.